So hello students, welcome to Aakash again. And you know from the previous session, we are here to discuss AIATS plus phase 2 paper, that was mock test 7, that was conducted on 2nd of March, that 2025. And in this session, we are going to discuss those questions where most of the students have made frequent mistakes, okay? And for the session, I am going to discuss the questions that belongs to these topics, that is sexual reproduction in flowering plants, organism population, plant kingdom, morphology, cell the unit of life, and photosynthesis, okay? So let's start the session. So the first question on the screen is question number 92, that is from plant kingdom, and what is common between pinus, cedrus, ginkgo and cycas. So all these plants that are mentioned here belongs to gymnosperms, and you know gymnosperms consist of seeds but they lack fruits, right? And all these particular plants have certain features. Let's discuss these features so that we can find out what are the common features present in all these plants, okay? So first we will discuss about pinus, cedrus, cycas and ginkgo, okay? Now first we will talk about the root system of gymnosperms, okay? So if you see the root system, you will find that in general gymnosperms consist of taproot systems, and there are certain symbiotic associations that are present in some plants. Like if we consider root system, so all of these plants consist of taproot systems, okay? But there are certain symbiotic associations, like in case of pinus you will find mycorrhiza, in which fungi, some fungus forms a symbiotic relationship with the taproot, and this will also enhance the productivity of water absorption, phosphorus absorption for the pinus plants, okay? While in case of cedrus, it's a taproot system, okay? In case of cycas, you will find another kind of symbiotic association where bacteria are involved, or you can see eubacteria like cyanobacteria are involved for making a symbiotic relationship with the taproot system, okay? Where in cycas, the symbiotic association is also called as forming coralloid roots, okay? These roots looks like corals, right? That's why the name coral or coralloid roots, because if you see the system or the root system of cycas, you will find that they have very much peculiar similarity with the corals that are found in the oceans, okay? So that's why the coral or coralloid root term has been mentioned. Now in case of ginkgo, it have taproot systems, okay? Now apart from this features, you will see the stem structure, okay? Let's move to the next session, that is stem structure. So in case of pinus, cedrus and ginkgo, you will find that the stems are branched, okay? So there is branched structure, branched stem, here also branched, and in case of ginkgo as well, here branched, okay? While as in case of cycas, you will find that the stems are unbranched, okay? Now let's move to the another section, which is leaves, okay? So if you see the leaves, in case of pinus and cedrus as well, you will see that the leaves are needle-like, okay? Why do they do this kind of modification? So you know gymnosperms are able to adapt in a dry tropical regions, and as the atmosphere is quite dry and a lot of heat is present, so leaves modify themselves into small pointy structures consisting of cuticles, okay? So they will prevent the process of transpiration in these localities or regions, okay? So in case of pinus, it's needle-like leaf, okay? Same here. Okay. While as if you see the cycas, you will find that the leaves are pinnately compound leaves. What are pinnately compound leaves? So yes, in these leaves you will find that the main leaf lamina is divided into certain segments, right? the lamina touches the midrib of the leaves, and this will lead to compartmentalization of the leaves into smaller leaflets. Okay, so these are called as pinnate leaves. So here they have pinnate leaves. Okay, and in case of ginkgo, the leaves are fan shaped structures. Okay, now in gymnosperms in general, you will find that the male gametes are quite reduced, and they are called as pollen grains, and these pollen grains are pollinated by air currents. Okay, and what happens is that pollens that are released from the sporophylls will transfer by the air current to the mouth of the megasporophylls. Okay, and there the process of pollination will occur. Now for all this structure, for all these plants, the main common feature is the pollination agent. Okay, while as if you see the other structures, what is other common features, that is they have taproot system, but there are certain symbiotic associations that are present in case of Pinus and Cycas. Okay, so let's see the question. What is common between Pinus, Cedrus, Ginkgo and Cycas? Yes, so absence of branch stem. No, branch stem is absent in case of Cycas, but in all these plants there is branching, right? So this option is incorrect. Presence of coraloid roots, yes, coraloid root is present in case of Cycas, not in case of all these plants. So this option is also incorrect. Formation of protonema, yes, what is protonema? Yes, protonema is a gametophytic structure that is found in mosses. Okay, and mosses belongs to bryophytes, not from gymnosperms. So this option is also incorrect. The fourth option says transfer of pollen by air currents. So this option is true. Okay, so for this question, the correct answer will be option number 4. Okay, so let's move to our next question. Yes, now next question is of sexual reproduction in flowering plants. Okay, the diagram that is mentioned here is a pollen grain. Okay, it is a mature pollen grain which consists of two cells, that is vegetative and generative cells. Okay, so the labels are mentioned here, and they have given certain options which can be correct about the mentioned labels. Okay, so let's try to understand the structure of pollen and how they are produced in angiosperms. Okay, so pollen grains are formed by the sporogenous tissues. Okay, so sporogenous tissues. Okay, these sporogenous tissues are made up of compactly arranged cells, right? And each cell are potential pollen mother cells. That means each cell is able to produce pollen grains. Okay. Now, the process of formation of sporogenous tissue cells into pollen grains is called as microsporogenesis. Okay. Okay. Now, here you will see that the pollen grains, in most of the angiosperms, you will find that 60% of angiosperm, in 60% of angiosperm, you will find that the shedding or the dehiscence of the pollen grain carries out in two cell stage consisting of vegetative angiosperms consisting of two cell stage which comprise of vegetative cells and generative cells. Okay, so let's see the structure of a pollen grain. Okay, so the structure that is given here is mentioning about label A. So label A is a exine. So yes, pollen grains are divided into two layers, that is outer wall layer and the inner wall layer. The outer wall layer is called as exine, which consists of sporopollenin. Okay. Now this compound sporopollenin is very much hard, okay, and also it is resistant to various alkalis and acids. Up till now, no enzyme or any chemical is known that can degrade this sporopollen, okay. Apart from this, exine have certain morphological features, right? You are seeing this structures here, right? This is also a peculiarity about exine, that they have certain morphological structures, as well as you will find that certain portion of exine lacks sporopollen. These portions are called as germ pores, okay. They are called as germ pores, as they lack sporopollenin. Now, what's the purpose of a germ pore? Yes, during the process of pollination, the pollen grain that are attached on the stigma will now form pollen tubes, and the pollen tubes needs to be released out of the exine, and the portion from where they are going to be released is from a germ pore, because they are not able to break the sporopollen, which is quite hard, right? So that's why these germ pores are there. Now, below the region, below the exine, you will find structure C, that is intine. Now, intine consists of thin cellulose and pectin consisting membrane, which is present below the exine, okay. So, cellulose and pectin comprising structure, okay. Now, you are seeing two different cells, one is quite large and other is quite small, okay. The larger part or the larger cell that is mentioned here as B is a vegetative cell. Now, vegetative cells comprise of irregular shaped nucleus, right? These two structures are formed due to the process of asymmetrical mitosis, okay. Now, here the nucleus is irregular, okay, and the cell itself is quite large as compared to the generative cell, okay, and its main purpose is to store the food and provide nutrition for the germinating pollen tube, okay. So, it comprises of food material or enrich in food material. The other portion that you are seeing, which is this, which is like spindle structure, right? This cell is spindle shaped, right, where the middle portion is quite broad and the narrower or the tip portions are quite narrow. So, here this structure is generative cell, and generative cells, as I have already told previously, that it is spindle shaped, comprise of nucleus and consist of dense cytoplasm, okay. So, now all these details has been mentioned on the board, right. So, let us try to solve the question that was asked in the previous slide, okay. Refer the given diagram and identify the parts labeled as A, B, C and D. Yes. So, A is a exine, right. So, first option is incorrect here. Let me use another pen. First option is exine, okay. A is exine. B is intine. No, B is a vegetative structure or vegetative cell. So, this option is incorrect. Third says it's an exine, okay. B is vegetative cell, true. C is intine and D is a generative cell. So, this option is true. The fourth option says that A is intine, which is incorrect. So, for this question, our correct answer will be option number 3, okay. Now, let us move to the next question. So, the next question is polysome is. So, this question is belong to cell, the unit of life, and polysome. This word is made up of two names, that is poly. Poly means multiple and zome means here refers to ribosomes, okay. So, polysome, it is also called as polyribosome. Okay. Now, what are these polyribosomes and polysomes, right? They are nothing but a structure consisting of mRNA. This is an mRNA, okay, which comprise of multiple ribosomes. On a single strand of mRNA, there are multiple ribosomes, okay. Now, in case of prokaryotes, you will find that these ribosomes are 70S types, okay. While as in case of eukaryotes, you will find they are 80S types, okay. If we talk about prokaryotes here, or 80S type, if we talk about eukaryotes, okay. Now, why do they need to make this structure like a single mRNA consists of multiple ribosomes? Yes, it's profitable in case of prokaryotes. If we talk about, it's profitable because a single strand is now able to synthesize multiple chains of amino acids, okay. Multiple enzymes will form from a single mRNA, so it's quite efficient. Similarly, these structures are not membrane bound organelles, they are just present in the cytoplasm if we talk about the prokaryotic cells, okay. Now, and if you talk about eukaryotes as well, it is present in cytoplasm, okay. So, this was all about the discussion of polyribosomes. Now, polysomes is a membrane bound organelle. No, it's not a membrane bound organelle, yes. Second option says several ribosomes attached to a single mRNA to form a chain. Yes, that's the correct option. Third option says subunits of ribosome. No, it's not a subunit of ribosome, though the subunits are attached on a single membrane or single mRNA to form polypeptide chains, okay. So, this option is also incorrect. Fourth option is composed of RNA and lipids. No, it composed of RNA, mRNA specifically, and ribosomes, okay. So, here this option is also incorrect. So, correct answer will be option number 3, okay. Let's move to the next question. So, the next question is from cell itself, cell the unit of life, and there are certain structures that are mentioned in the column 1. While as if you see the column 2, there are certain features about this column 1 structures, okay. So, let's try to understand each and every structure that are mentioned here in the next slide, okay. Now, you have seen bacteria, right? Under the microscope, you are able to see a bacteria. Now, this bacteria is rod-shaped structure. So, the terminology that we are going to use for this bacteria will be bacillus, okay. Now, this is the cell wall. Now, it consists of cell membrane, okay. It's a cell membrane. Sometimes you will find that cell membrane have certain inclusions inside the cytoplasm of a bacteria. Now, these structures are called as mesosomes, right. Similar type of inclusion bodies are also found in case of cyanobacteria, where they consist of pigments that are called as cytochromes, okay. Now, apart from this, you will find certain cell surface structures, like here you will find long structures that are called as flagella, okay. Now, flagella comprise of filament, hook, and basal body. So, if you zoom out this structure, you will find that there is outer membrane, there is peptidoglycan layer, and then comes the inner membrane, okay. So, let me draw the diagram here, okay. So, in the outer membrane, you will find the L size or L clip, right. In here you will find rods, and in the peptidoglycan you will find P, and then you will find S or MS, and below this you will find C. Okay, so these are the complete anatomical structure of a filament. Above this structure you will find a hook, and then the flagella, which is made up of flagellin protein, is present here. Okay, but that's not the point here. What's the point here? That the flagella is the outermost portion which is responsible for the motility of a bacteria. Apart from this, there are other certain structures that are present in a bacteria. That can be a pili, okay, these are pili, or can be fimbriae. Now you will see that I am drawing fimbriae very smaller, while as the pili are quite long, and you will find that fimbriae and pili, they both does not provide any support for the locomotion of a bacteria. Okay, so they do not carry out locomotion, while as the flagella is responsible for locomotion. Okay, the pili and fimbriae are specific structures. In case of pili, you will find this structure in the fertile strain of bacteria, F+ strain of a bacteria. What does its function is that it provides conjugation. That means they will transfer some amount of DNA to its other bacteria which is compatible to it. Okay, while as it's like a sexual reproduction, but it is not truly a sexual reproduction. Okay, while as if you talk about the fimbriae structures, these fimbriae structures are helpful for a bacteria to stick to its substratum or to a host tissue. Okay, so these are the functions of a fimbriae and pili. Sometimes you will find that certain bacteria, if we talk about purple and green, blue and green bacteria, you will find certain gas vacuoles inside the cytoplasm. Okay, these gas vacuoles are responsible for providing the buoyancy to the cell. Okay, so they can float on the surface of a water or surface of any medium. Okay, so these are called as gas vacuoles. Okay, so this was all about the structure of a bacteria. So let's try to answer the question that is mentioned here. Okay, so fimbriae helps to attach the bacteria to the host tissue. Okay, while as chromatophores contains photosynthetic pigments. Okay, and plasmid, here we have forgot about the plasmid here. So plasmid is an extra chromosomal genetic material that are found in case of bacteria, and if we talk about the yeast cell itself, there is also plasmid there. So what's the purpose of a plasmid? Yes, plasmid provides certain phenotypic character to a bacteria. Let's say a plasmid consists of a gene or a protein, okay, gene that is coding for a protein, okay, and that protein or enzyme is going to digest the chemical which is responsible for killing a bacteria. Let's say penicillin. So some bacteria are penicillin resistant. That means they are having a plasmid consisting of a gene that are coding for an enzyme that will degrade the penicillin. Okay, so that's what the feature is given by the plasmid. So it's like a blessing to a bacteria to survive in a harsh condition where other bacteria are not able to survive. Okay, so here plasmid provides resistance to antibiotics. Gas vacuole helps provide attachment or provide buoyancy to the bacteria. Okay, so A matches with 2 here. So A matches with 2, this option, and the fourth option is incorrect. B matches with 4, so this option is also incorrect. Here B matches with 4, C matches with 1, and D matches with 3. So our correct answer will be option number 3. Okay, so let's move to the next question. Okay, so the next question is from morphology of flowering plants and the features or in Solanaceae family flowers have. So in Solanaceae family you will find that the flowers are actinomorphic, which is represented by plus sign, right? The flowers are bisexual, okay, and they comprise of calyx that are 5 in number and fuse. This condition is called as gamosepalous condition. Okay, Then comes corolla, which is also 5 in number and fuse, that is gamopetalous condition. Now the androecium is present also in 5 and it is attached with the calyx. So this is epipetalous condition here, and then the gynoecium, which consists of bicarpillary syncarpous condition. So this is the condition, and the gynoecium are superior. That means we will underline this gynoecium. Okay. So that's what the flower floral formula gives us a lot of information about a family. So let's see, in Solanaceae family, flowers have epitapelous condition? No, it have epipetalous condition here. This is epipetalous condition, not an epitapelous condition. Okay. Epitapelous condition is found in Liliaceae because there is no certain division between a calyx and corolla. They are fused and forming a tepal or a perianth condition. Okay. So this option is incorrect. The second option says it's an actinomorphic symmetry. Yes, the flower is an actinomorphic. This symbol represents actinomorphy. So this option is true. Third option says unisexual flower. No, the flowers are bisexual. This symbol represents sexuality of a flower, which is bisexual flower. Okay. So this option is also incorrect. The fourth option says that it's a monocarpillary ovary. No, there are bicarpillary ovary. They have bicarpillary ovaries and they are superior. Okay. So this option is also incorrect. So here correct answer will be option number 2. Okay. I hope this concept was clear and you are able to identify each and every symbols that are represented here. Okay. So let's move to the next question. Select the odd one out with respect to sexual decide seen in Ophiris and bee. Okay. Now, you have generally seen a honeybee or a bumblebee pollinating a plant, right? Basically, this interaction is mutualistic, right? Pollen is now transferred to other plant for pollination, right? So plant is getting an advantage as the pollination process is carrying out, and also the bee is getting nectar, right? But there are certain plants which has evolved around a time and they have developed certain features that are quite similar or are responsible for attracting a particular insect. Okay, some of its example is a Ophiris. Now it's an orchid. Okay, you can see the structure of a orchid plant. Yes, you are seeing this structure. Now this structure that you see looks like a bee. Okay, specifically a female bee. It's a female bee like structure. So in orchids or in Ophiris, if you see, what will be there, that one petal will transform into a female bee like structure. Okay, so what is now carrying out the pollination process? What happens? The female bee, or the male bee that is represented here, is now going to move or perceive it as a female bee and copulate the female. Okay, but here as the this process or this phenomenon is called as pseudo copulation. Right, the male bee is perceiving this petal which is looking like a female bee as its own, okay, species, and it will copulate, but this process is false. So this is pseudo copulation. Now due to this, what happens, the flower or the Ophiris is now going to dust its pollen on the surface of this male bee, and again the male bee will move to other Ophiris perceiving this as a female bee and again try to pseudo copulate that Ophiris, and due to this the pollination process will occur. Here the male bee is not getting anything, that is no nectar that is present or given by the Ophiris. What it's presuming it's that it's a female bee that is going to be a partner of its life, okay. But sadly, it's a pseudocopulation, or it's a false representation of a female bee. So Ophiris has the advantage that it gets pollinated, but the male bee is not getting any advantage here, okay. So that's what the representation is said here, okay. So let's see the options. Select the odd one out with respect to sexual decide seen in Ophiris and bee. So the organism exhibit this type of interaction are also show coagulation. Yes. What will happen if this structure that you see is not evolved in a certain manner where it resembles a female bee? Yes, the rate of pollination will be decreased, because now the female or the male bee will not perceive the petal as a female bee, and the process of pollination will be deprived, okay. So this structure that you are seeing of a single petal develop into a female bee like structure is a evolutionary process, or it is a co-evolution process where it is trying to resemble a female bee of a particular species of insect, okay. So this option is true. All petals of flower bear an uncanny resemblance to male bees. No, they resemble female bee. And also all petals are not responsible forming an uncanny resemblance with a female bee. Only one petal is able to form an uncanny resemblance with the female bee. So this option is incorrect. Pseudocopulation can be observed during the interaction. Yes, pseudocopulation is observed. Whereas this strategy is adopted for pollination. Yes, the Ophiris has adapted this process so that it can pollinate itself, okay. So this option is also true. Here the correct answer will be option number 2, okay. Now let's move to the next question. Identify the incorrect match, okay. So now this question belongs to photosynthesis, okay, and photosynthesis specifically about the C4 plants or C4 pathway. There are certain comparisons as well. If we talk about this question, there is certain comparison that is C3 plant and C4 plant. So let's compare the C3 plant and C4 plant so that we can identify the correct solution about the question, okay. So in case of C3 plants, you will find that the primary carbon dioxide acceptor is a 5-carbon molecule, okay, and the 5-carbon molecule here will be RUBP, okay. Okay, it is a 5-carbon molecule. Now if we talk about C4 plant, the primary carbon dioxide acceptor which is present in the mesophyll cell is a 3-carbon molecule which is phosphoenol pyruvate, okay. Next is the first stable product. If we talk about the first stable product in case of C3 plant, so you will find that the Calvin cycle, the first product is a 3-carbon molecule which is called 3-phosphoglycerate, okay. And if we talk about C4 plants, then the first carbon product or the first stable product is a acid which is oxaloacetic acid, okay. Rubisco is present in mesophyll cell in case of C3 plants, while as if you compare this with the C4 plant, the rubiscos are present in the bundle seed cells, okay. Now what are bundle seed cells, okay. So there is certain anatomical difference in case of C4 plant. You will find that the leaves consist of vascular bundles surrounded by bundle seed cells. These cells are compactly packed cells forming a compact structure where no intercellular space is present, and the cell's peculiarity is that it consists of thick-walled cells. Due to this structure, the atmospheric gas, okay, the atmospheric gas is not able to percolate inside the cytoplasm of this cell. Okay, so in atmosphere, you know, oxygen, carbon dioxide and other gases are present. So, and you also know that Rubisco here, that the Rubisco favors carbon dioxide if the carbon dioxide and oxygen concentration in the environment is equal. It will favor this. But if oxygen concentration is quite large as compared to carbon dioxide, then it will carry out the process of oxygenation. And this process is you can say not fruitful to a plant, because what it does, it break down the fixed carbon dioxide into 3-phosphoglycerate and 2-phosphoglycolate. Okay, so it's a wasteful process. That's why what happens in C4 plants, there is certain modifications where bundle seed cells are present, and the bundle seed cells have a certain mechanism where the atmospheric carbon dioxide is coming from the atmosphere into the mesophyll cell, okay, and the mesophyll cell transfers this fixed carbon dioxide into the bundle seed cell through a plasmodesmatal connection. Okay, due to this mechanism, the amount of carbon dioxide present on the enzymatic site of Rubisco, okay, on the enzymatic site of Rubisco, carbon dioxide is quite large. That's why the process of photorespiration is very negligible. Okay, so that was the difference between a C4 and C3 plant. Let's move to the next. Calvin cycle. So Calvin cycle occurs in C3 plant in mesophyll cell. Okay, while as in C4 plant, this process occurs in bundle seed cells. Okay. The special features that is seen in case of C4 plant is this anatomy, which is called as Krenz anatomy. Okay. There is other special features, like if you talk about the presence of chloroplast, you will see that in bundle seed cell you will find the chloroplasts are agranal. That means the grana, or sorry, thylakoids are not stacked in a grana. They are free. And the abundance of chloroplasts is quite large in C4 plants, bundle seed cells. Okay. The optimum carbon dioxide concentration that is required to a C3 plant is quite high. Okay, it's about 450 microliter per liter. Whereas if you talk about a C4 plant, you will find that the concentration of carbon dioxide that they require is very low as compared to the C3 plants. Okay. Now, if atmosphere comprise of global warming, right? Now the temperature is quite high and the amount of carbon dioxide is getting even more. What will happen? The C3 plants, which is requiring a large amount of carbon dioxide, will able to adapt that environment because the carbon dioxide concentration has been higher. So it is quite positive for a C3 plant to able to survive on that global warming level. But if we talk about C4 plant, as the carbon dioxide concentration has been increased due to the global warming, consistent high concentration of carbon dioxide will damage the process of photosynthesis in case of C4 plants. Okay, so that was all about the difference between a C3 and C4 plant. I'm not going on in detail about the process of Calvin cycle and Hatch and Slack pathway. Okay, so let's see the question. So maize exhibit double fixation of carbon dioxide. So maize is a C4 plant. Okay, maize is a C4 plant, and in C4 plant you will find that the mesophyll cell will take up the atmospheric carbon dioxide into a soluble form and then again release this carbon dioxide in bundle seed cell for the process of Calvin cycle. Okay, so there is double fixation occurring in a C4 plant. Second option says bundle seed cells in C4 plants have Rubisco enzyme. Yes, so bundle seed cell in C4 plants have Rubisco enzyme, which is correct. Third option says primary carbon dioxide acceptor is RUBP in sorghum plant. Now sorghum is a C4 plant, and in C4 plant the primary carbon dioxide acceptor is phosphoenol pyruvate, not RUBP. Okay, ribulose bisphosphate is not the first or primary carbon dioxide acceptor here. So this option is incorrect. Fourth option says oxaloacetic acid. So yes, oxaloacetic acid is a first stable product in case of C4 plant. So first stable product of carbon dioxide fixation in C4 plant. So this option is also true. So correct answer will be option number 3 for this question. Okay. Let's move to the next question. So the next question is, choose the odd one with respect to stem modification. Okay. So you will find that there are certain modifications adapted by a plant, whether it can be a modification of a stem, modification of a leaf, right, modification of a root, right. These adaptations are basically for a plant to survive to its environment or to provide certain support or protection to a plant. Okay. So if you talk about the modifications that are mentioned here, let's see the modification one by one. Okay. So tendrils of watermelon. These structures are developed by the axillary bud. Now, the axillary buds develops themselves into slender, thin, spiral structures, okay, which provides support to a growing stem. Okay. So, it's a modification of stem. Okay. The second option is phylloclade. Now you have all seen a cactus, right? You have seen the spines on the cactus and the green portion of a cactus, right? So let's draw the diagram of a cactus. So this is a opuntia, right? This is a opuntia, right? Now you will find that the opuntia consists of spines, right? And the bulgy structure or the flat structure that you are seeing here is a stem. Okay. So this structure is called as phylloclade. Now the plant adapts itself into such form because the leaves are now converted into spines to reduce the process of transpiration. As they are present in a xerophytic condition, the stem modifies itself into a green photosynthetic structure which will now perform the process of photosynthesis instead of a leaves. Okay. So this structure is called as phylloclade. So it is a modification of again a stem. Okay. Third option says tendrils of pea. So in case of tendrils, if we talk about peas, so these structures are modification of leaves. Leaves modify themselves into slender, thin spiral structures which will now provide support. So this is a modification of leaves, not a stem. Okay. So here the odd one is the tendril of pea. Okay. Fourth option is thorns of bougainvillea. So thorns of bougainvillea is again a modification of axillary bud or a modification of stem. Okay. So here our correct answer for this question will be option number 3. Yes, Roman, your answer is correct. Very good. Now let's move to the next question. So the next question is again from the morphology, and quite a times NEET have asked about the floral formula of a particular plant, right? And you need to remember the plant from which belong to. Okay. So in case of this question, belladonna belongs to, yes, Solanaceae family. Okay. And in case of Solanaceae family, you will find certain floral features like the inflorescence is solitary, axillary, or cymose, as in solanum. Okay. Apart from this, if you talk about the sepals or the outermost wall of a flower, this is calyx. Okay. So the calyx consists of 5 subunits or 5 sepals which are united and so valvate estivation. कौन सा? Valvate estivation. Okay. Similarly, if you talk about the corolla, Yeoman, these questions are from AIATS plus phase 2 paper that was conducted on 2nd of March. Okay. So calyx, then corolla. Corolla are also 5 in number. That means petals are 5 and they are also united, and they also show valvate estivation. Okay. Now, if we talk about the Solanaceae family Androecium, you will find that they are 5 in number, okay, and they are free and fused with the petals. Yes, so they are showing epipetalous condition. Okay, so they are showing epipetalous conditions. Okay, if we talk about the Gynoecium, so Gynoecium here is bicarpillary syncarpous. Bicarpillary syncarpous, and the placenta is swollen, and it consists of axial placentation. Okay, the flower or the gynoecium is located above the thalamus. All the other floral parts are below it, so it's a superior ovary. Okay, so that was all about the feature of a Solanaceae family. If you talk about the seed itself, the seed is endospermous and many, while as if you talk about the fruit, then it's berry, or you can say a berry-like fruit. You have seen a tomato, right? So that's a berry. So here you will see the floral formula of a Solanaceae family is comprised of actinomorphic flower. Okay, they are bisexual. Okay, then they consist of calyx, corolla, androecium, and gynoecium. Androecium is fused with the corolla. So here the correct answer will be option number 2nd here, while as all these are from other, that is, this is from Liliaceae family. Okay, okay, this is from Fabaceae family, and this is from Brassicaceae. Okay, so here the correct answer will be option number 2. Okay, let's move to the next question here. So the next question is of resource partitioning mechanism. Okay, so certain times you will find in nature there are various competitions occurring between a species, right? If we talk about, let's say, a species A and another closely related species which is species dash. Okay, these two species have the same food resource. Let's say there is a fruit, let's say apple. Okay, now the resource is apple and they both need to eat an apple. Okay, here they will compete for this same resource, right? And what will be the result? If A is here, that is mentioned, is superior. Okay, superior in case of strength, superior in case of food eatability, right? Easily consume high amount of food, then you will see after certain while the A dash will extinct, right? This concept is called as competitive exclusion. Yes, Raimon, you have said correctly, right? It's a competitive exclusion. And this concept was given by Gauss, and he said that in a limited resource region, if a two species that are very closely related will compete for a particular resource, then the superior one will be the one which will survive, while as the inferior one will be extinct. Okay, but in nature that's not the case here, because resource is limiting though, but there are certain interspecific competition as well. Means, let's say we will bring another species, okay, B, in this diagram. Okay, Now bee is also able to eat apple. Okay, it's able to eat apple as well. Okay, so here what will you see that there is competition between the two species as well, two very closely related species as well, and also different species or intraspecific competition as well. So it's not going to be in case that both these species are now going to extinct. In fact, this species will coexist by partitioning the resource, okay, or the behavioral pattern where on the time which will they eat an apple, right? If it is eating an apple at 1 a.m., then it will eat an apple at 6 p.m. let's say, and it will eat a food like let's say 2 p.m. okay. So that in this way they are able to survive on a same resource and without any competition or without any in between competitions. So this will lead to coexistence of this all organisms, okay? Coexistence. And this concept is called as resource partitioning, which was seen by Mark Arthur in case of warbler birds, right? He have seen that in case of warbler birds, he found that there was a tree, okay, and this tree comprised of different closely related warbler birds that are located in specific location. Let's see, this one species was present at quite top of a tree, one species was present at a middle portion of a tree, and one species was present at this portion. Due to this partition, they are not now competing for a same resource, right? They are present on their own niche, and now they are carrying out their life processes, okay? Now if we talk about the competitive release, right, this statement or competitive release, Connells have done an experiment in which he have gone to Scotland and find out that some large and superior barnacles that are called as balnus, okay, in Connell's experiment he found that large and superior barnacles, here it's a balnus, is present in a intertidal area. Okay, so let's see the concept here. Let's see. So this is a rock, okay, and here you will find an ocean. Okay, now you will see that balnus are quite populated in these localities where intertidal zone is present. Okay, why they are quite large and superior, whereas if you see Clathamellus, inferior one, is present above this region or intertidal zone. So in presence of balnus, the Clathamellus are not able to grow themselves in the intertidal area. But what will happen if I remove the balnus from this region? What will be the scenario? Yes. Now, what will be the scenario here? That as the superior species has been removed from the intertidal area, the Clathamellus will now try to grow themselves in this intertidal zone as well. So this is a competitive release. That means the superior species has been removed from its habitat, due to which the inferior species is now able to relocate or diversify itself in different localities or different regions. Okay, so this is a competitive release. Okay, I hope Riomon, your competitive release concept was clear here, right? So let's see, resource partitioning mechanism promotes coexistence. Yes, it promotes coexistence. It promotes competitive exclusion. No, it does not promote competitive exclusion. Okay, it promotes competitive release. No, and it promotes amensalism. What is amensalism? Yes. Okay, so in amensalism, the basic example that you see in case of interaction between a penicillin and a bacteria. Okay, so what happens? A fungus which is called as penicillium releases a chemical which is penicillin, and due to this chemical release, the other bacteria that are present in the locality are not able to occupy that niche or get died due to the process. Okay, so this is called as amensalism, where one species is getting all the areas, while as here you will see that in case of amensalism, one species is not getting any effect, right? Penicillin produced by the penicillium is not having any positive effect on a penicillium, right? But if you talk about the other bacteria, it's getting destroyed or getting killed by the penicillin, okay? So this type of interaction is called as amensalism, okay? So that was all about the concept of resource partitioning, okay? I hope the concept was clear to you for today's session. I hope for the next session you are going to see the zoological concepts in which we will talk about a lot of concepts from the zoology. So I hope your NEET exams will be better, and also stay hydrated and read NCERT for future sessions, okay?